Namaste students, I am Jayoti Dr. Rajesh Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's Institute, Jaipur. Today I am going to discuss about the topic microorganism of extreme environment. So this is the continuous part of microorganism of extreme environment. In the previous video I discussed about some organism of extreme environment. So today I am going to discuss about the Archae and their classification system. Some biologists have organized living things into large groups like archaebacteria, eubacteria, protists, fungi, plantae, and animalia. These six kingdoms were classified. But in 1996, scientists decided to split Monera. Okay? They decided to split Monera into the two groups of bacteria, archaebacteria and eubacteria. Because of these two groups of the bacteria were different in many ways, scientists also created a new level of classification which is called the domain. So we have three domains in Monera. The first one is the bacteria, second one is the archaea, and third one is the eukarya. The kingdom archaea, if we talk about the kingdom archaebacteria, any of a large group of the primitive bacteria having unusual cell wall and membrane lipids, ribosomes, and RNA sequences, and having the ability to produce methane and to live in the anaerobic, extremely hot, salty, and acidic environment are the organisms which are known as the archaebacteria. So the organisms which have unusual cell walls, unusual membrane lipids, and they have unusual ribosomes and RNA sequences, and they also have the ability to produce methane. Okay, and they live in the anaerobic environment, and they live in the extremely hot, salty, and acidic conditions are known as the archaebacteria. These archaebacteria are the ancient bacteria, and some of the first archaebacteria were discovered in the Yellowstone National Park, Houston. And the prokaryotes are structurally simple, but biochemically they are complete. Some basic facts which need to be known. So the archaebacteria live in the extreme environment like hot spring, salty lakes, and normal environments like soil and ocean water. And they are unicellular, and there is no peptidoglycan in the cell wall. Okay, and some of these archaebacteria have a flagella that is that is used for their movement, and most of them don't need oxygen to survive. They are anaerobic in nature, and they can produce adenosine triphosphate from sunlight. Like some of them are phototrophic microorganisms, and they can survive enormous temperature conditions which normally bacteria cannot survive, and they can survive under rocks and in ocean floor, the ocean surface, and they can tolerate huge pressure differences. The, these are the characteristics. Now talk about the structures of archaebacteria. The size of archaea are slightly less than 1 micron long, and a micron is about 1/1000 of a millimeter. And in order to see the cellular features, scientists use powerful electron microscope. And if we talk about the shapes on the of the archaebacteria can be spherical, it can be ball shaped like coccus. Okay, and there are some also round shaped bacteria which are long, thin, and labeled bacillus. And a variation of cells have been discovered in square. Locomotion some archaea have flagella, hair-like structures and assisting the movement, and there can be some one or many attached to the cell outer membrane. Protein network can also be found on the cell membrane, and these these structures allow cells to attach on the different type of surfaces. The cell features within the cell membrane. The archaea cell contains cytoplasm and DNA, which are in single-looped forms called plasmids. And archaea, most archaea cells also have a semi-rigid cell wall and helps it to maintain its shape and chemical balance. Some of these structures protects the cytoplasm, which is the semi-liquid gel that fills the cell and enables the various parts of the function. About the phospholipid, the molecules that make up cell membrane are called phospholipid, which act as a building block. In archaea, these molecules are made up of the glycerol, ether, ether bonding. What is ether bonding? The ether bonding makes it possible for archaeobacteria to survive environments that are extremely acidic or alkaline or that have extremes in the temperature. So this is the feature by which these archaea can survive in extreme environment like phospholipid vitae and ether bonding. If we talk about the classification of the archaea, there are five phylum: Crinarchaea, Crinarchaea, and Euryarchaea, Coraarchaea, uh, Thumarchaea, and Nanoarchaea. So these are the five phylums of Archaea. The example of Crinoarchaea is one of the best characterized membranes of the Crinoarchaea is Sulfolobus sulphoreticus, which is isolated from geothermally heated sulfuric springs in Italy and goes at 80 degrees centigrade in pH of 2 to 4. Next phylum, the Euarchaea, diverse with seven classes: Methanococcus, Methanobacteria, Halobacteria, Thermoplasma, Thermococci, Icobaclovi, Echinopyrae, and they consist of 9 orders and 15 families on the basis of habitat, and they are divided into following methanogens, extreme halophiles, sulfate reducers. Halophiles are the organisms which can survive at high salt conditions, and the methanogens are the organisms that produce methane and metabolic byproduct in an anoxic condition. And thermophilic organisms are the organisms which can survive at high temperature, and sulfur reducing bacteria which have the capability to reduce the sulfur. Next class is the Coracoita, and they are derived from the Greek noun koros, which means young man or young woman, okay, which means ancient. Mm, they are also known as the Gina acuta. They are found in the hot spring in the Kamkata. Next class is the Thomo acuta. The Thomo acuta are proposed in 2008, and after the genome, the symbiosis was sequenced and found to differ significantly from other members of the phylum. And all organisms of this lineage are identified as chemolithotrophic, ammonia oxidizer may play important role in biogenomical cycles. Next class is the nanoarchaea. In taxonomy, the nanoarchaea is meant by the old dwarf. They inhabit high temperature environment with an optional uh, growth of 90 degrees centigrade and are highly unusual because they grow and divide on surface of another archaea. Nanoarchaea which were discovered in 2002 contain both the smallest known living cell and the smallest known genome. Members of phyla have been detected in whole cultures. Now, what are the importance of the archaeobacteria? The first one is the extremophilic enzymes. The extremophilic archaea are resistant to either to heat or to extreme of acidity and alkalinity. What are the uses? Thermostable DNA polymerase such as the PFU DNA polymerase from Pyrococcus furiosus are used in the PCR and amylases, galactosidase, polyoleinase, and other species of the Pyrococcus that function at our 100 centigrade allow food processing in the high temperature. Um, the archaebacteria are also used in the sewage treatment. Some methanogenic archaea carry out anaerobic digestion and produce biogas extraction of metals. Some acidophilic archaea extract are used in the extraction of the gold, copper, and cobalt from their ores. And some methanogenic archaea can decompose, grow in biogas fermenter, carved into methane gas. Some now role of archaea in chemical cycles. Some of archaea have important role. They play important role in carbon cycle, nitrogen cycle, sulfur cycle, etc. And they help in the research and the ability to. This is due to the ability to extreme conditions, which help researchers learn about the climatic conditions. Similarly, also have important role in production of some antibiotics. So this is all about the archaebacteria and what kind of questions can be asked. The question which can be asked from this lecture: explain archaebacteria and their morphological structures and what is their different uh, classes. So I hope the students will better answer all these questions. So my lecture is over here. If you have any query, you can ask me. So thank you very much.